defense and their abilities to fend off stress and disease. But I will start with just 2 or 3 sides of the bad news. The reason that folks like me and our lab are employed is to handle some of the challenges with beekeeping and with bee colonies themselves. And we're especially focused on the biological challenges, the parasites and pathogens. And beehives are vulnerable to those because they're a pretty good environment if you're a virus. You have this packed. This room's a few people, but you can imagine probably 1,000 of us in here all mingling and passing food with our mouths and flying out to flowers that themselves have viruses and parasites on and bringing it back to the family. So in that environment, bees are exposed to lots of diseases that they then, in large part, they themselves have to fend off. The beekeeper can do something, but not so much. Then, of course, for many of the beekeepers who are our stakeholders, our customers as researchers, are also carrying out their beekeeping on a big scale. They're moving bees across environments and at high densities across the country, ending up not so far from here for pollination events like the almonds, where the bees are in some cases a little bit nutritionally stressed, but also as a sort of disease studying person, they're exposed to each other's diseases in many respects, and that's a way to rapidly move we think new viruses around the country and such. And nevertheless, bees are still around. There's not all colonies like this in our apiaries now. Most of the colonies we get probably about like here, maybe 70% overwintering survivorship that they make it through. And we think mostly because those colonies aren't exposed to the worst of the worst, like a combination of diseases, and they can fend them off. And yet some of them, we know, and partly because we do it on purpose, in some colonies we're giving them, we're not treating for mites, we're challenging them with viruses, we're really throwing a lot at them, and they still survive, they still make it through. And that's, again, in large part from their own innate defenses, their abilities to fend off these diseases. And we can help that, both as bee breeders, and there are many folks in this audience who are doing this, who are improving the genetic stock of bees, and also through management. So these are the topics I wanted to talk about. I'm going to talk about briefly those parasites and pathogens that we're most worried about, and then ways that we can take genetic insights of bees and of their reproduction to understand how they themselves in many ways take care of a good fraction of the problems. So for the bad actors, the diverse parasites and pathogens in the bees, there's a number of viruses, mostly RNA viruses. The closest equivalent to us is maybe poliovirus or hepatitis A virus, or they don't have a DNA stage. They're purely an RNA-based species that in bees at least are really good at reproducing. They're also, unfortunately, good at moving into honeybees via vectors, especially varroa mites, down on the bottom left. And so we have that sort of left side with the viruses and the mites is really the big target for helping bees, to trying to reduce those impacts. And then, of course, there are bacterial diseases and some fungi. And how do bees fend them off? Well, they actually do a good job on their own. They have through millennia, because they have defenses. They have behavioral defenses. You've heard of hygienic behaviors, the ability to recognize disease and help their nestmates to fend off disease. What we're interested in is also immune responses, so that once a bee does lose the roll of the dice and gets an infection, they're able to decrease the impact of that infection and actually remove, clear the infection from their own bodies. And they're I'll show you a couple stories with that. One that we've done with American foul brood disease, and this wasn't, in some sense, this is a way to maybe to develop a better stock of bees. But for me, the most fun thing about this is we found with run-of-the-mill Italian honeybees from Georgia that we could find traits within them, really good resistance or immunity traits towards this disease, and that some of that actually transfers to other, we think, to other infections. And this is just a quick sort of our again, I'm going to become less and less of a beekeeper in many ways and more of an indoor biologist. But we have to study these diseases in sort of a controlled environment. And in this case, we take first instar larvae from comb, and then here is their new home, their only home actually after that, which is to raise up in an artificial diet. But we can challenge them then with an infection and then follow their responses. And so we know in this, on the right are sets of 24 32 bees each from different lines of bees. We can challenge them with bacteria, see who survives that challenge, and then also look at some of their traits to get at that way. And this is what it looks like, which is actually really fun. Not only did we show that they had an immune response, they actually, like us, they responded to infection, but that response could be bred. It was a heritable response. It's something that, in this case with artificial or instrumental insemination of bees, we could push that trait into bees through their dads. They had a genetic trait that came into them, and it actually did help them survive. A really cool study, and this is actually from a colleague who's also largely doing lab work, but he had the wisdom to collaborate with some beekeepers in Indiana, Greg Hunt, with a group of bee breeders, and they said, okay, you know, you can do your lab work on the side, but we're going to try something that's a little more closer to a trait that we can measure as part of a breeding program right now. And they measured across colonies in their breeding stocks for what they call mite biters or ankle biters. And these are colonies in which the hygienic bees not only picked mites off each other, but they took those mites that they picked and actually damaged them. They bit into the mites and did damage. And the predominant trait, the best kind of selectable trait, was actually pulling the legs off their mites that they were doing, which is so these are vicious bees, but they're clearing the mites, grooming each other for mites, but they're also damaging those mites so they don't get back into the system on another bee, and in so doing they're able to reduce the mite population. The really cool thing about this particular trait, and this is again without any not really understanding the genetics or doing tests for the genetics, strict line breeding of highly functioning bees or queens, daughter queens whose sisters had shown this trait. And then by taking that trait, they actually forced upward this behavior of biting the mites to something like 40 or more % of the bees. So they could actually get lines of bees in that selection process over, this is 6 years, I guess, drive that trait into the population to a really high rate. And what's interesting about this, and really surprising for most genetic traits, is that it's actually ramping up. Most of the times with selection or with breeding for a trait, you get an initial success, so you kind of, okay, you're doing better, and there's an asymptote. It actually runs out of steam after a few years in the case of bees. They're actually getting a positive return. They're getting more of this trait as time goes by. What he's doing now, and actually Mia has researched this as well, that their plans with this program is they're trying to actually deliver these queens back to the field. So they've selected the trait, deliver them back paired with non-selected queens, and actually see in the field if these are good bees, if these are a stock of bees that a beekeeper would want to manage. And also if the trait, more importantly in some sense, does this trait hang in there over the next generation, maybe with one bit of outcrossing, which is a worry with many selectable traits or desirable traits, is that the shelf life, as it were, of them is one generation only, and after that you have to back up and go back to the pure stock. But this is really exciting. I do have, if it works, sorry. Yeah, sorry. I'll do that at the end. I have a little video of showing the hygienic behaviors, which you've seen probably in your colonies. The ability of the bees to both, in many cases, honeybees can recognize mites on their sisters or even on themselves. But in these selected bees, they're really vigilant. They don't give up. They know there's a mite there, and they actually keep crawling around their nestmates until they get that mite. And then, of course, in this breeding line, They don't just pick the mite and let it fall to the bottom board. They actually do some damage to the mite all the way. The next kind of interesting avenue of understanding the colony life of bees, but also the mating of the queen, has to do actually two traits of the male, of the drones, of the males in the colony. And these were actually discovered 160 some years ago, that the males are haploid. So the males carry one set of chromosomes, one version of each gene in each of their cells, whereas we carry two in each of our cells, as do the female bees. And that has two important implications. One is for those of us doing breeding studies with bees, we've got a really great way to kind of mainline in a genetic trait, right? Because if the males are genetically identical and we can screen that male for a good trait, resistance, we can put it into a colony or into the next generation much more efficiently than in other systems. And we do it by artificial insemination, but we score the males and then actually cross them into a line and can increase. In this case, this was a study with resistance traits, again, from Germany, where the actual, again, the breeding power, the power to move in a good trait, is much higher because the males are haploid. They're a stronger way to get them in. The other neat avenue that's been looked at for mating in bees has to do with diversity within the colony. And you're probably, I think Tom Seeley probably mentioned this, and in this understanding that honeybee hives are made up of this really a good genetic mix of different lineages. Because the queen mates many times, you have these subfamilies going on with different patrilines. So most of any pair of workers in a colony are probably half sisters with each other as opposed to full. And those traits carried by those different, from those different dads, actually lead to a better functioning colony, a better, as Jerry mentioned, a better superorganism for the colony. And what's interesting about it is that kind of makes sense for some behaviors, right? Bees that are, maybe you get a mix of bees that are a little more hygienic and bees that are less hygienic, and you can, kind of mix those traits together. It seems to also work for these immunity traits, the disease resistance, and that's what we're still not sure of a mechanism per se for that, but we know that the colonies with a diversity of males, so a diversity of sperm stored by these queens, actually tend to have higher productivity and survivorship. This is one study looking at, again, artificially inseminated queens with a pool of diverse males versus one male. Survivorship, on the top bar, that survivorship of the diverse family groups is much better. In fact, only those colonies in this particular study survived. It's much better over winter, and it seems to be something about just having that diversity. Another recent study by David Tarpey and also Jeff Pettis in our lab looked especially at disease, and they're in that case, the brood area and population sizes were lower with the single drone colonies, and there's strangely a higher level of disease resistance in those. And that, again, we think of disease resistance, or I was thinking of it as a pure genetic trait, that you've got it or you don't. There's colonies, lineages that have disease resistance and not. What they were able to show is even for the same gene pool, the same set of genes, the bees whose moms mated with many males actually tended to have lower disease on the whole. So there's something about the group effect of having these perhaps resistance traits that keeps down disease. And that's something that, again, is doable within a breeding program with the stock that you have, is to maintain pretty high matedness of the queens. Another avenue that's sort of, again, the sort of organism side of the colony, or even beyond the level of the bees, is we're starting to see with genetic tools, especially, that there's a whole set of microbes that bees carry that aren't these bad actors that I mentioned. There's bacteria and fungi that are actually doing positive things in the colony. And the analogy that comes up is similar to our own gut bacteria as humans, that's involved with digestion, perhaps disease resistance. We're starting to find those traits in bees, And people are thinking of ways to apply that to actually make beehives a bit healthier. And this came about in part, it was through the technological advances. We knew there were bacteria in bees, but we couldn't actually see them very well. And nowadays we have tools to follow where these bacteria live. These are larval bees, kind of a cross-section head to toe of a larval bee, and then one right through the middle. We know where the bacteria are living, and we also know when particular ones are there, if that results in a bad fate or a good fate. This is one of the bad ones. The green in the middle of these bees is Paenobacillus larvae, again, the American foulbrood bacterium. But we also know that in those larvae, there are bacteria that aren't causing disease, and we started to wonder if some of those might actually be beneficial. And this was driven in part from experience where you can inoculate larvae with the pathogen, Paenobacillus larvae. It's highly virulent, and 10 or so spores can get larvae sick with foulbrood and die, be removed from the system. But many times in doing that, even with 1,000 spores or more, the larva doesn't die. And so we were puzzling over that. Is that an immune response, or is it something else? And sort of by serendipity, we found that in many cases when they did survive, they had other bacteria that were hanging out in those larvae, and we think kind of doing a battle against the bad bacteria. So they were somehow inhibiting the American foulbrood bacterium. And these studies were carried out, again, largely in the lab, but also trying to mimic this in the field, to put back these good bacteria and see how potentially sick larvae would do. And we're on a 4,000-acre campus, so we actually have an apiary that's quarantined, but in which we're able to study American foulbrood disease. So we have many colonies that are really filthy with it, and which is a good thing in our case, so we can do stuff. And this is that apiary. What we're able to do is to take colonies, inoculate the brood with the pathogen, and at the same time inoculate it, and some of those same larvae with different species of bacteria, and follow them. This is sort of on the ground here, but that's basically a clear sheet of plastic where we painted on color coding for each cell of each different type, and we scatter them over the brood to kind of randomize them a bit. And in these experiments, it wasn't pretty. I wouldn't say that nobody's going to go out and clear American foulbrood by this approach, but we could follow them as they went through different treatments, different other bacteria to add in, and look at development, see how well they survived. And consistently, across, I think now 16 or 17 different trials, we find lower disease in the ones that also got some other species of bacterium. And the yellow-colored ones are ones that got an additive of the same amount of the pathogen, but a little bit of, in this case, a Brevibacillus, another bacterial species. And so the yellow and orange ones are consistently higher across these trials, and another set here. It's not, again, we're not clearing disease, unfortunately. We thought we maybe, if it was really inhibitory, it might actually eliminate or clear the disease. But consistently, we're finding a better prognosis when they have this other species in there. This kind of led to some work with adult bees, and adult bees haven't been looked at with bacteria, in part because there aren't any known bacterial diseases in adult bees, or common ones at least. There are some sepsis events, but there's nothing that's really a threat, as it were, to adult bees. Nevertheless, adult bees carry tons and tons of bacteria. They have them throughout their intestinal tract, and so the question was, what are those bacteria doing? Are they actually just kind of commensals in there eating nutrients in the bee but not doing anything positive or negative, or are they possibly doing a beneficial role for the honeybee? And this is again an image inside a bee of two different species of bacteria, and you can actually see there's a large amount of bacteria growing in the guts of bees. So they clearly have them. The bacteria are thriving in there. It suggests perhaps that the bee is at least providing an environment whereby bacteria can grow, and maybe that is again a sort of mutualism, the bacteria doing well but also helping the bees. this work, and largely because we now have sort of genetic names for these, or genetic traits for them, has led to the naming of now actually 4 different groups of bacteria and bees. And it's sort of nice, these are named after sort of researchers and luminaries in the honeybee field. Robert Snodgrass, Martha Gilliam, who was actually a USDA researcher in Arizona, looking at these questions in a different angle, in bee bread especially, and Carl von Frisch. So these are the sort of the 3 of the main actors in the stomachs of bees that we really hadn't recognized, or no one had looked at much until we had genetic traits to be able to follow them. And given that, and given the culture of these bacteria, we can start to do experiments to see how they're affecting bee health, and ultimately to see if there's something that we want to have in bees or not, or something to control. We've done a series of these controlled experiments. Again, we actually pull uncapped bees as they're ready to eclose, pull them out, and then feed them with the good actors, we think, and then often another parasite or pathogen. And we've actually had, we have a benefit of having a local high school that has a science and tech program, so we've had a number of high school interns helping with these projects, and they make nice projects for them because we can choose one pathogen and then sort of do a series of experiments for their projects. We also have several people in the lab who are looking at different disease. This is Ryan Schwartz, who's a scientist interested in the trypanosome parasite of bees, Crothidia. It's another gut parasite. He's especially looked at how that and Nosema interact. But added to that, the interactions between that parasite and one of these native bacteria. And what this is showing is that, again, it's not, it certainly doesn't make bees immune from disease, but bees that are fed this commensal bacterium, this gut bacterium, actually have lower levels of the parasite when you also challenge them with this parasite. So this is really exciting in the sense that there is some sort of interaction or inhibition between these two. An even more interesting result came for viruses, actually, and this is really recent. A summer student in the lab, undergraduate student, discovered this, and we started off with just a correlational study. Just when we found high levels of deformed wing virus in bees, those same bees tended to have lower levels of one of these bacteria. We thought, well, it's probably just by chance. There's some other factor that's affecting these two. And so the way to tease that apart is to experimentally challenge them. And again, this is our native form of beekeeping, sadly enough, for these projects. Again, we established bees, and this is really because we wanted to keep them clean for these parts of the experiments. We established them inside and feed them one or the other of the parasite and the bacterium. In this case, actually, it was with viruses, deforming virus, and this is where collapsing months of work down to one little chart is only exciting in the sense if you really squint and look close. But that particular bar right there is showing that in bees that we had fed the virus and this bacterium, there was 0 virus at the end of it. So they actually had cleared off the virus that otherwise we thought would have been in there. And we found that before. And so the only way this can happen, the bacterium is not going after the virus. In fact, it doesn't really come in contact with it because the viruses are in the cells of the bee. But the bacterium seems to trigger an immune response, and that immune response helps the bees survive the virus. What we're doing with that part now is actually getting a bit into the field with it, getting the bees exposed to these 2 factors. We paint them any number of colors, so we have, in most of these experiments, we have about 16 different groups of pairs of microbes that we look at. Paint them those colors and put them back out in single box colonies. And then the fun part, after usually 7 days or 10 days, is we go through each of those colonies and basically with this mini vacuum, we pull out each and every painted bee that survives, so we can actually give them somewhat of a natural juvenile period growing up, but we've treated them first and we've followed them, and so we can measure survivorship of those bees. And then, for the sake of these experiments, we can actually measure the disease level, so how much virus and such do they have. And we found with these experiments, but in the case of the virus study, we haven't found a difference in survivorship. We're not inducing viral mortality in this case, but we have found the same result, that those bees that got this boost of the gut bacterium had lower virus at the end of the trial. So what does this possibly lead to, or any potential leading to for beekeepers? One is there is some viability, some possibility of doing an additive of bacteria for colonies. We know that these bacteria are common across colonies, but they're not in each and every colony. So maybe there's some value. And the top ones were lifted off the web for the sponsored Google search results for Walmart and such for human probiotics, but there's likely to be some benefit in beehives, and not taking yogurt bacteria or things like that, but actually taking the native bacteria they have, but making sure they all have the right amounts of those. And the other corollary to that is that it may be that our use of antibiotics, except in cases where it's really prescribed, really to clear an apiary of risk of AFB, post symptoms in another colony, where it's really needs to happen. If we do it prophylactically, if we do it as can be the case in the long-term nature, we're probably limiting these good bacteria as well, and there's evidence to that so far. So maybe the one management tool would be to kind of keep down that usage when possible. Okay, now I wanted to talk about some research in our group, but also in the community on kind of connecting the dots with different factors of bee health and bee disease, and hopefully giving and adding to the wealth of knowledge from beekeepers about how to manage disease in a sort of more holistic sense. And one of the exciting avenues lately has been on pollen and on nutrition. Judy Chen in our lab has collaborated with the USDA lab in Arizona on nutritionally supplemented bees being better resistant to viruses. So they actually get, with nutrition, especially supplements, are often used simply to build up colonies, to spur brood production, to get them going. But you can also get an added benefit that bees in those colonies, again, for the mechanisms aren't 100% clear, but bees in those colonies are less susceptible to virus infection. So we're trying to look at that. Is that something innate to the pollen? Is it true for any protein supplement, or is it again a sort of indirect effect of the immune response? And there's some really neat work with that, as is the work looking at types of pollen. So are different pollen classes better suited for bees? Is it better to have a diversity of pollen? Intuitively, people think, well, there should be a diversity of pollen, but it really hasn't been mapped out as well, especially across different habitats, that multifloral pollen or diverse pollen is unanimously better for bees, and that's a topic of great interest right now. This particular study from France actually did show that, and again, on a disease level, it's showing an impact to these pollens, certain pollens, on nosema, on an actual disease load. So we've known that pollen nutrition, that a balanced availability of diatropes helps production, helps the mass of rearing new bees. It also seems to connect into this immunity, to their ability to fend off diseases, and so it's kind of really exciting. Again, it's a way of, and again, maybe the bees are choosing this as well, but a way of kind of maybe medicating the hive, helping it through a rough time of disease exposure with a better diet. The other avenue that many people are involved with right now, of looking at bee disease kind of from above, as it were, is to do survey work and actually try to measure all the traits about beekeeping and bee risks that might impact their survivability. And this is, unfortunately, this is enabled by really high colony losses, right? So these are studies that, that are built on bad news, as it were, built on the fact that many states in the U.S. are losing 40 to 50% of their bees annually. In this survey by the Bee Informed Partnership, we have really substantial colony losses, but within that, there's potential, of course, to to detangle the environments of those bees and what they're exposed to, what they're facing, and see which of those risk factors are actually most important for disease. This is one study. Actually, it's a self-reporting study by beekeeping over their what the evidence is to the beekeepers suggested were causes of declines, from queen failure on the left to starvation, mites, nosema, and so on. And this study, we're starting to break this down by different categories of beekeepers, from backyard to sideline and commercial. And so the USDA in particular has historically, or at least for decades, really just measured colony numbers and colony fates of beekeepers with beyond many of us, and I have 3 hives at home, and many people do. They really want to measure the bigger operations in apiaries, which is completely valid for the industry, but it misses.
You have colony collapse disorder down as a cause of death. I thought it was an indication of death.
That's really a good. Yeah. It's a syndrome, so it is an indication, but it's something that's definable as a and you'll see that it's kind of tucked on the right next to "don't know." so these are this is trying to get a best it's a multiple choice test, as it were, for the beekeeper. So you have to put something that you would perceive, and it is a definable trait. I will say, colony collapse disorder has maybe been overused in some cases, but it really is this sort of syndrome of the worker bees vanishing, as it were, from the scene quickly with the queen intact without any obvious mite problems, is at least something that's definable. While you're quite right, though, it's more of a phenotype or a symptom than a cause. But I think it's a valid thing to measure just because it is so unique from some of the other causes. And in this case, it's not the prevalent reported cause of bee declines, and we've known that for some years now, that CCD is in the background of other causes of declines. It is recognized still by beekeepers. We think it's not completely gone to the winds, but it's not the major reported cause of declines. We have, and again, there's biological causes and pesticides in there as well. So we have kind of the range of reported causes, and the nice part about these surveys, if they're consistent and the wording is consistent, is they can be followed year to year, and we can actually see changes in areas and over time. And that's really, I don't pretend to be someone who's up to date on the pesticide risk or the risk of other factors besides disease. But if there is a risk, it should fall out in these sorts of studies, in the sense that bees in areas where the regime of crop treatment, say, change, would either show higher losses or a symptomatic change in those losses that would map that. So I think these sorts of data, for whatever favorite hypotheses people have, these types of data are going to help really provide more solid evidence for what's going on. And again, the sad news is they're built on the fact that bees are still declining. We're still getting 30-some% losses, overwinter losses alone in the U.S., and about 50% on a 12-month cycle. So colony losses are still quite high. They're higher than they've been in other parts of the post-Varroa era. So there's something going on that can do that. There are also these surveys, and actually, if you really want to, spend a night looking through, statistical data and numbers, they're starting to post really neat data, decoupling this by feeding treatments, geography, and all these kind of factors that somebody, maybe hopefully even somebody outside of the research world, because people get kind of tunnel vision on some of this, but somebody hopefully will compile this data and actually see patterns that help in managing disease. It may be a different pattern in different areas, but there are data now. There's a really this is a sort of a $5 million survey that's going on right now, and they're generating lots of information that hopefully will help beekeepers, and hopefully will fairly soon, in the next couple of years, be a way to kind of make sense of the field patterns of losses. And this is one just showing different forms of sugar supplements that are involved there. And the surprising result that despite sort of intuitions of some people, different treatments such as high fructose corn syrup in this case and sucrose did not seem to lead to significantly changed levels of survivorship. In fact, beekeepers who were feeding their bees back frames of honey in some sense had more decline to date in the survey than those using maybe more artificial carbohydrate sources, which is a surprise to me, I thought. Another survey that's gone on in parallel has actually been focused on sort of production beekeeping, but in a stationary apiary. So we're trying to just establish the same genes, the same genotypes of bees, and this runs counter to some of the interesting work lately with locally adapted stock, or stock that might be best in different parts of the world. But what we decided to eliminate that genetic component and just drop bees out of the sky from one genetic source and see how they did over time in different environments. And those environments, simply by virtue of where many of us lived, were set up as follows in these 7 states. And so this project ran for several years, starting with package bees from Georgia, so from the southeast, and establishing them throughout the country. And the point here was to then take any bit of data we could about those environments, from climate data for that year to just the land cover and the usage, the crop usage around those sites, and just to try to see, it's limited in the sense it's not the whole scope of beekeeping. The California apiaries, for example, I think we're all in Southern California, not up here, because Kirk Fisher, who was at Riverside, was the locus for doing that in California. But it's taking those sites, measuring traits around the apiaries, and then just looking over time to see what happens to the bees. And again, we can get sort of climatic changes of the bees. We had a variety of regions sampled and trying to get maybe a diversity of climates for the bees, and certainly different levels of agricultural usage, and we think a density of beekeeping. Again, as with this nationwide survey that's still ongoing, this study, sadly in some ways, in both incarnations of it, had a lot of colony losses. We basically lost, half of the colonies within a year in the first case, and then almost all of them in the second case by the end of the study. So pretty high colony losses, and again, a lot of kind of material for trying to understand what those losses reflected. One of the results that was looked at was people measuring different climate events during this process. Not a big pattern, nothing that really struck that seemed to map with colony losses. Here's a little trend, possibly with supersedure rates and warmer climates, but it wasn't so exciting. As with most studies, there was also this non-biological component of looking at the usage of pesticides and crop cover around these sites. As with many beehives, there was pesticide exposure of these bees, and they brought them back. In this case, it didn't fall out differently enough across the sites in the sense that we could correlate that with colony losses. But there were some events actually with supersedure, especially, where there's kind of starting to be borderline trends of, you see in 2009, pesticide diversity. Basically, the gross number of pesticides had some level of variation with supersedure. Not the primary correlate by any means, but interesting. And then we have the bad actors again, and the colonies that weren't able to fend off disease. So we're trying to say most colonies are able to survive in the face of disease, but some of them weren't. And the ones that declined, we find are to me, really interesting. And again, it's more something to think of the defenses of the colonies, the abilities to fend off disease. What we saw on the right here, those bubbles, the circles represent the loads, the copies of viruses and other diseases in the bees, and they get a bit bigger. The blue ones on the right for those 2 viruses plus Nosema apis were significantly more. So colonies, weaker colonies in decline, had more of those 2 viruses, and they had more Nosema apis than did surviving colonies, strong colonies. So those individuals were higher. All of the parasites, ironically with the exception of IAPV, one of the viruses, all of the rest were at least slightly higher, trending higher in the sick colonies. And then what's really crazy, and we call this sort of a pathogen web, is those dark black lines. And what that means is you can think of it sort of like a food web. It's like all the interacting species in this case inside the bees, affecting them. We're going up and down together. So basically, you would find in declining colonies often several viruses that went up all at once, or 2 viruses and Nosema apis, or 2 and Nosema cerani. And so the dark lines are kind of the lines of connections between those, where those particular bad agents, especially the viruses, would kind of go up together in the sick colonies. And so what we think is happening, there's 2 things that could be happening. One is it just reflects that those bees are generally weak. They have a poor immune system. They're not able to fend off anything that comes their way. And then the second factor, possibility, is that certain viruses interact with each other. And it's kind of sort of like the good bacterial story, but with 2 bad actors, that when 1 and 1 of the bad actors are added together, it equals 3 or 4. There's a worse effect by adding these together than predicted. And so we're thinking that those, some of those particular viruses are especially kind of toxic pairs together, and trying to find out what that means. Is it that bees that are less, have a poor immune response to one, similarly have a poor immune response to the others, or is it that one really sets them off on a bad path whereby they can't fend off other disease? And that, again, we're doing experimentally by giving them different doses of viruses, and we're also trying to understand how the bees could fend off some of these viruses, and if it's a breedable trait, if it's something we can really improve in their abilities, or if they can improve themselves with a little bit of help from us humans. I wanted to acknowledge our group in Beltsville. This is the gang. There's really about half of us are sort of paid civil servants there, and the rest are visitors. We have a lot of students coming in and out, and visitors from different countries who do projects with us, which really amplifies stuff, and it makes it a lot more fun. In the lab that we have, there are 5 scientific groups. Besides myself, there's Dr. Judy Chan, who's doing virus work, and again, also is doing this sort of virus and nutrition work. Jeff Pettis is doing, right now, focusing largely on queen health, trying to look at supersedure, the impacts on supersedure of both disease and pesticides. Stephen Cook is there. He's actually a toxicologist, so he's looking at the physical changes in bees when they're stressed by chemicals. And then Miguel Corona is trying to integrate nutrition and longevity, so especially with worker bees. If they're given a better nutrition as larvae or as adults, do they then survive better, function better as foragers and worker bees? So that's, I'm going to stop there, but I'd be really happy to take any questions you might have. Okay, there you go. So yeah, this, I'll narrate. It does have a little music going with it, but we don't need that. So watch this little cluster of bees right here. And this one in particular has a mite on her, so she has something going on. She's not being super proactive herself to get the mite off. She's just waiting and waiting, but her nestmates, especially this one, are relentless. They're just trying to find that mite and to remove it. So it's sort of a heightened grooming or hygienic behavior. They won't stop, in a sense, until they find the mites. And you can see, I mean, if you would do, if you watch bees with mites on them, or even put them on the mites, there are some cases where, I mean, the mites are very good, right, of slipping under the plates on the backs of the abdomens of bees. They can hide for a while. But there are some cases where the bees know there's a mite there and sense it on themselves, or if they've recognized it elsewhere, and they just won't give up till they pick it off. And so I think these are bees where that's more prevalent, where they can they will keep going till they find those mites. And in Greg's case, in that study, they find the mites, and then as a extra punishment, they pinch them a bit with their mandibles, or they bite their legs off. Yep.
My eyes are beginning to fail, unfortunately. But when I look at my bees, sometimes I notice a color that's the color of a mite, and it's right between the thorax and the abdomen.
Okay.
And I often wonder if it's a mite, and if mites have a tendency to go right in there, because I'm not that familiar with the anatomy, right in there because it's so tiny. And they get right in there, or where are they found in that location between the thorax and the abdomen?
I think when they certainly will circle through there. When I see them when they're really hiding out, it seems like they get back on the abdomen and kind of wedge themselves between the plates. Yeah,
so not
hard
like right
plates.
there, like right in that
Yeah, I
tiny
don't know.
spot.
I mean,
I see this glimmer of varroa mite color, but I can't see the
There's a
varroa
hiding
mite.
spot,
But I
yeah.
normally can see the varroa mite, and I just would think that half of it's hidden. Maybe other
Part of
people.
it's there. Yeah, I don't know. Do people? Is that a notorious hiding place for mites to be between the abdomen and the back of the thorax? I could see it. It might work for them.
Do you have anything to say about the effect on the health of the bees generally of the percentage of the of drones in the hive? Because Dr. Seeley says the bees like 1 in 6 to be drones, but beekeepers apparently insist upon keeping it down to 1.06. And most of my experience of other people's hives is that some of the energy of the bees goes on to honey production, the rest goes on to a struggle between the beekeeper and the bees as to how many drones they can have. Particularly every time they take the hive apart, they kill all the drones that are being raised, and next week they come back again, and that happens again. So what is the effect of this battle, and should the bees win or the beekeeper?
It's a very good question. I think discretion is the better part of valor. I will not add anything to what Dr. Seeley has mentioned on that. The question was relating to the ratios, perhaps an optimal ratio of drones to workers in colonies versus what by management they achieve. And I'm sorry, I don't have any insights whatsoever into that. Yeah.
You had several charts that were looking for patterns, and by looking, and it was all across the country. And the one thing that for me seemed to be lacking was an overlay or a correlation with Africanized areas versus non-Africanized.
Okay.
And it seemed to me that since the traits and characteristics of Africanized bees are so different in so many ways, that may have an effect on the data that's not or maybe looked at as a factor.
Yeah, it's a good question. So the question is, as Africanized bees become more prevalent and ingressed into other lineage of subspecies of honeybees, are we starting to see any changes in survivability or in these sorts of studies? And certainly in areas that are highly Africanized, their losses, I would say, tended to be lower, certainly to mite losses because of a higher ability to withstand mites. But I don't know. This particular study, we didn't measure Africanization. And in fact, actually, the stationary apiary study, they were all Italian bees from Georgia. So that was one element where the genotype of the bees wasn't the same. For the National Honeybee Survey, though, it's a bit more just, landing in a certain geography and sampling bees, and I'm sure there has been some sampling of Africanized bees in that study just by the numbers, but they haven't been distinguished genetically from the other ones. So, so the only bit on disease in Africanized bees and maybe colony losses has been from researchers in Southern California and in Tucson, simply having mixed apiaries and also having apiaries with Africanized bees and not treating them and finding better survivorship with the Africanized bees in those cases. So they do have beneficial traits as well as the defensiveness traits in swarming. Yeah.
On the status on the importation of genetic materials from the USDA, has that been any changes?